Here you see the drops in energy as the electrons pass down the electron transport chain from FMN to Q to B to cytochrome C1 to cytochrome C and cytochrome A, and then eventually to oxygen. And the red arrows indicate those drops in energy that are sufficient to drive the production of an ATP molecule. So and once again you can see that you get 3 ATPs for every reduced NAD and 2 ATPs for every reduced FAD. Now this slide shows you what would happen if you were to block the electron transport chain. And what would happen is simply this. You can see there at the red stripe, there's a blockage. The electrons can't pass from cytochrome B to cytochrome C. The result is everything sort of backs up from that point, and you get to the point where you can't add the electrons from NAD into the electron transport chain. As a consequence of this, all of the NAD in the mitochondria and ultimately in the cell will be trapped in the reduced form, which means, if you remember, that you won't be able to run glycolysis because the glycolysis requires the presence of oxidized NAD. So if all of the NAD is tied up in reduced NAD, glycolysis cannot run, and if glycolysis cannot run, you die. Here you see the effects of 5 poisons on the electron transport system. Notice that rotenone blocks transport of electrons right after the addition of electrons from reduced NAD. As a result, electrons cannot pass any further down the chain, and because they can't pass any further down the chain, they result in an absence of the ability to make ATP. Cyanide and carbon monoxide, you can see, bind a bit farther down the chain, and they prevent further electron transport down the chain at that point. As in the previous slide, in both of these cases, with rotenone, cyanide, and carbon monoxide, everything backs up from that point onward, actually from that point backward, and the result is that glycolysis can't run because all the reduced NAD is basically, all the NAD is tied up in the reduced form, and consequently there's no oxidized NAD, glycolysis can't run, and you die. I should point out that both cyanide and carbon monoxide were used by the Nazis to kill people in gas chambers during World War II. Carbon monoxide was used first, and it turned out that they were wasting too much gasoline producing the carbon monoxide to kill people in that way. Consequently, they moved to cyanide, which was cheaper. Okay, the next poison is something called dinitrophenol, DNP. And what DNP does is it essentially allows hydrogen ions to cross the membrane without going through ATP synthase. And as a result, you basically dissipate the hydrogen ion gradient, the electrochemical gradient of hydrogen, without allowing it to be used to make ATP. And the result is you burn through sugar a lot because you keep trying to build up that gradient, but you don't use it to make ATP. Now, this was actually used, I think it was in the 1930s, as a weight loss technique, because if you use this, you would end up losing lots of weight. And it was working for a while, but then it turned out that it was also killing people because they couldn't get enough ATP to survive. So they stopped using it as a diet technique. I don't recommend using it in that way. Oligomycin, finally, if you look at what oligomycin does, it is blocking the transport of hydrogen ions through the ATP synthase. Consequently, you can't make ATP. And of course, if you can't make ATP, what happens? Let me hear it. You die. This is a schematic diagram of the components of the electron transport chain within the inner mitochondrial membrane. Take a few seconds and you can look at it. This is another schematic diagram of the components of the electron transport chain within the inner mitochondrial membrane. It's simply rendered in a different way and gives you some different information, but it's basically the same thing. This is an ATP tally sheet, and we're going to go through it. and look at how many ATPs are made at each stage of glycolysis, Krebs cycle, and respiration. When you look at this, this is going to be the summary of the energy yield from one glucose molecule. You have to keep in mind that both NAD in its reduced form and FAD in its reduced form donate electrons to the electron transport system. You have to keep in mind that each reduced NAD made yields 3 ATPs from the electron transport system, and each reduced FAD made yields 2 ATPs from the electron transport system. First, remember that in glycolysis, you make 2 ATPs as a final yield of ATP from glycolysis. And remember, that includes, and that's a net gain. You used up 2 ATPs, but you then made 4 for a net gain of 2. Also keep in mind that during glycolysis, you make 2 reduced NADs, and each of them can translate in the electron transport system into 3 ATPs apiece for a total of 6 ATPs. However, 2 ATPs must be used to transport electrons across the mitochondrial membrane, so the result is that you have a net gain there of 4 instead of 6. That's 6 minus the 2 ATPs needed to move electrons across the mitochondrial membrane. And so there you'll get a net gain of 4. It turns out, however, that some cells have figured out a way to get these electrons into the center of the mitochondrion for free, and so those cells can squeeze an extra 2 ATPs out of this, and I'll talk about that later, but it turns out that cells that can do this are cells that have a high energy requirement, cells, for instance, like liver and kidney. Next, 2 NADs are obtained during the on-ramp to Krebs cycle, where pyruvate is converted into acetyl coenzyme A. Those 2 reduced NADs can enter the electron transport system, and you will get 3 ATPs apiece from them for a total of 6 ATPs. Next, I hope you'll recall that during each turn of the Krebs cycle for metabolizing one complete glucose molecule, one turn for each of the 2 3-carbon fragments that originally came from glucose, you get 1 ATP apiece per turn, so that's a total of 2 ATPs from the Krebs cycle itself. Next, recall that 6 reduced NADs are produced by the 2 turns of the cycle required to break down the full glucose molecule. These 6 reduced NADs are each converted into 3 ATPs for a total of 18 ATPs. And also recall that from each turn of the Krebs cycle, of the 2 turns required, you get 1 reduced FAD molecule, and reduced FADs, remember, are converted into 2 ATPs apiece for a total of ATPs produced from FAD of 4 ATPs. If we add up all these ATPs, the 2 from glycolysis, the 4 net gain that we get from the NAD reduced that are produced during glycolysis, the 6 ATPs from the conversion of pyruvate to acetyl-CoA, the 2 ATPs from the Krebs cycle, 18 ATPs from the NADs produced by the Krebs cycle that go into the electron transport system, and the 2 FADs from the electron transport system. If we add that up, we get 36 ATPs. In the case of those highly energy requiring cells that have figured out a way to get the electrons across the mitochondrial membrane for free, then we get an extra 2 ATPs for a total of 38 ATPs. This slide shows you how polysaccharides, fats, and proteins relate to the Krebs cycle and glycolysis, and where they feed into the process at various times. Now, if polysaccharides are available, they're broken down into simple sugars. They go into the top of the glycolytic pathway, and they're broken down as they go through, and will eventually enter the Krebs cycle as acetyl coenzyme A. So if you have lots of polysaccharides on board, you will preferentially use those because while polysaccharides carry less energy than fats, they're much more easy to mobilize. However, if you run out of polysaccharides, you will then start breaking down fats. The fats are broken down into glycerol, the backbone of the phospholipids, and fatty acids are broken down into acetyl-CoA. Fatty acids are built up 2 carbons at a time by using acetyl-CoA if you run those arrows backwards, and when they're broken down 2 carbons at a time, with acetyl-CoA carrying the 2 carbons and having them enter the Krebs cycle at the normal place. Glycerol is broken down and it goes into the glycolytic pathway at the level of PGAL, and then that will also enter the Krebs cycle through pyruvic acid. Now, this is why it's so hard to lose weight, by the way, because in order to start breaking down fats, which is where we keep most of our weight, you have to run through all your available polysaccharides. Then you move to the fats. Now, if you run out of fats, now you have to start breaking down proteins. The proteins are broken down into amino acids, and they can enter either the Krebs cycle or various points during the glycolytic pathway. This is problematic because when you start breaking down proteins, you start breaking down things that you need, and this will eventually lead to death. In people who are starving, they've gone through their available polysaccharides, through their available fats, and now they're breaking down proteins. And a couple things is going to happen. Number one, if they're breaking down their proteins, they're going to eventually start breaking down proteins that they need. Also, when you break down amino acids in this way, you give off ammonia, and ammonia, if it builds up in too high an amount, is toxic. And so this is what happens to people when they are starving. As a lead-in to the next topic, photosynthesis, this slide shows you the connection between respiration and photosynthesis. Cellular respiration breaks down glucose using oxygen and gives off water and carbon dioxide. Water and carbon dioxide are then, through light energy, used to build up glucose, giving off oxygen as a waste product, and the whole thing goes around in circles like that, with the two things working in a complementary fashion. This slide serves as a summary of aerobic respiration, and it shows you the electron transport chain, how the electron transport chain relates to the Krebs cycle, the TCA cycle in the picture, and then shows you the hydrogen ions building up in concentration between the inner and outer mitochondrial membranes, and then shows you the hydrogen ions going back into the mitochondrial matrix through ATP synthase and being turned into ATP, using that energy and that electrochemical gradient to produce ATP by chemiosmosis. So take a look at this, and just to sort of get an idea of where things are happening during aerobic respiration in the mitochondrion. And as a final review, here's a respiration overview showing you glycolysis occurring in the cytoplasm, Krebs cycle occurring in the matrix of the mitochondrion, and oxidative phosphorylation, or chemiosmosis and electron transport, taking place in the inner mitochondrial membrane with the ATP synthases attached to that membrane. And when you look at it, you can see that ATP is produced in all three places. So take a look at this thing just to sort of give you an idea of what the overview is, and good luck.